So, those of you who attended last talk was about the structural brain network and the anatomical connections and how do we analyze the brain structure. And in this talk, I'll be talking about the brain function. Uh, how do we model the functioning of the brain once we have the structure? So, this is most from the current work which I've been doing for past few years at Dartmouth. And so, yeah, so what do you call a mechanistic model? Why do we need a mechanistic model? And there can be many purposes, there can be many questions you might be tackling, and based on that you choose what scale of modeling and what type of modeling you want to use. So if you're just interested in large-scale dynamical behavior of the brain, you can just use mass models like Wilson-Cohen or Jensen-Ritz and so on. But if you want a mechanistic understanding of what's happening right from the level of receptors to neurons to circuits, right into the behavior, and to make some predictions, what happens when there's a dysregulation of the brain circuit and some disease happens, or what happens when there's a drug intervention, then you need a multi-scale model, which can not just describe the system at different scales, but also address the causal relationship between these different scales. So, what happens at receptor levels affects the neural spiking, that affects the brain dynamics, that affects the cognitive state or the behavior. And there's always back and forth going on. So, we successfully have demonstrated that such a modeling is possible, and we can get very promising results from that when we modeled a corticostriatal circuit in the brain. And what is a corticostriatal circuit, for those of you who don't know? It's one of the very key circuits for the brain function, not just for the primates, but right across the animal kingdom, whoever have brain. So there's a cortex, which is the evolutionarily the latest structure in the brain, and the more complicated the animal gets, the higher you get in the evolutionary timeline, the thicker the cortex becomes, the more important the cortex becomes. And if you go down the evolutionary tree, if you go to primates or if you go to the rats, the striatum is what becomes more important. But together they make a very important unit. So caudate and putamen together makes striatum. Then there is pallidum, and there is thalamus, and it's a loop, it's a circuit. And there are various nuclei. There is brainstem, there is substantia nigra. This circuit is important in lots of cognitive behavior, like action selection, motor control, what you call reinforcement learning. And when it's a dysregulation of this circuit, there are lots of diseases like Parkinson's disease, major depressive disorder, even bipolar, and so on. So lots of psychiatric treatments target the corticostriatal circuit, and that's why it's very important. So this is the schematic of the model of that circuit. There is a cortex, there is a visual cortex, the posterior cortex, the anterior cortex, and the striatum. The striatum in reality is very small, but here it looks very big because that's how important it is in the model. So, a plugin. This is the larger project which I have been working on. It's NeuroBlocks. It's an open source software platform which we have been developing for computational psychiatry. And most of the models, the modeling approach which I talk about, are incorporated in this, and it's available freely. You guys can go and check it out. It has tutorials. It has a self-paced course where you can. So it's a modular modeling tool. So you can actually build neurons or brain regions and connect them and play with them. And it can also help you analyze the actual brain data with scientific AI tools. So, coming back to the model. So, as I said, there should be certain properties in the model which you want. It has to be multi-scale. It has to be biologically constrained, which means you cannot make too many abstract theoretical assumptions. You have to follow what we know from the physiology and the anatomy of the brain, what is connected to what. It should have the spiking neurons. It should have the spatiotemporal firing patterns, and it should have the brain regions at the level of brain regions. So the connections at the level of brain regions, and most of the behavior which you observe should emerge out of it bottom up, rather than being imposed on it. So you are just encoding the physiology at the neuronal level, the spiking, the ion channels, the receptors, and most of the high level dynamics and the behavior you observe should emerge out of it. And it comprises of something we call biomimetic computational parameters. So it is a theoretical construct, but it does exist in the brain. So instead of categorizing or dividing brain into brain regions or a neuron or sub-circuits, we can also think of it as being formed of computational parameters, which is, it could be a assembly of neurons, it could be a large population of neurons, any sub-circuit which performs a computational operation, and it is essential to perform that computational operation, and it's irreducible. You need the entire sub-circuit for that operation. And these are examples of some of the computational parameters that we have used in this model. And they are all from the existing literature. So individually, all of them existed in different literature, and we have to consolidate it into a single model. One of them, an important one, is this one, the local feedback lateral inhibition circuit. It exists in the cortex. So we might see most of the cortical models around there. They often treat neurons as a soup, like a random networks, all to all, or a sparse random network. But actually, it's highly structured, and the structure is important. What's the structure? So it's formed of local circuits, which are like this. There are pyramidal cells, and there are interneurons, which are called feedback interneurons, which are inhibitory, and they form a local circuit like this. So each of them is exciting their interneuron, and the interneurons giving a feedback inhibition to all of them. So what happens is, when all of them get an input, and you can see that there are different synaptic connections, for a given firing pattern, one of them or some of them will have a higher firing rate than the others, and that one will win, and then that will inhibit all others. So it's a lateral inhibition mechanism, also called winner take all, and something like this. So these are the pyramidal cells, and this is the interneuron, and you can see only one of them is firing. It ensures that it's all of, like, it's the one which with the highest input only is allowed to fire, and others are not, and that ensures that the cortical firing is not going berserk. And when you organize them, they are then your lateral inhibition feedback circuits are then arranged into a superficial cortex. So this is a sample of that.
So I just have a general question about this. I mean, I'm not too familiar with this. So I mean, is there like a sort of a reason for having like 4 such units? I mean, like so like you need like a minimum number of such units? I mean,
This one?
Yeah.
Yeah,
Just curious about that.
So this is how exactly that occurs in the brain, 4 to 5.
Okay.
The ratio is exactly that.
Okay.
So 4 to 5 pyramidal cells for each interneuron.
Okay. Yeah, sure.
Yeah, I mean, it's an interesting question. What happens if you change that ratio.
Yeah,
That's
I mean,
like a.
that's what I was wondering. I'm wondering like why is this motif like reoccurring? Or I was wondering what could be.
Yeah, yeah, that's something which can be found out, but it is how it is in the actual brain. So,
Sure.
so that's how.
Just I wanted to say that I gave the same explanation during my talk. It's typically in a cortical microcircuit. If you record from, let's say, primary visual cortex layer 2, 3, or 4, because you cannot actually look at all the brain areas. But if you do it at the population level or single neuron level, we have seen these during our recording at MIT as well, that the ratio of the excitatory neurons or pyramidal neurons, which Anand is talking about, and the inhibitory neurons are typically 3:1.
Alright, yeah.
The reason being, the inhibitory neurons have a firing rate which is about 9 to 10 hertz, whereas the excitatory neurons during spontaneous state generate a firing rate of 3 to 4 hertz.
Yes.
Because it is 3 times firing, to maintain the excitation inhibition balance, the number of inhibitory excitatory ratio being generally agreed to be like that. Yeah.
Thanks.
So once you have this model, we use a test case, a specific experimental data set to validate and check how much helpful that model is. So we use this specific behavioral paradigm called category learning from one of our collaborators' lab at MIT, Earl Miller, where they had many such experiments, but one of them consists of the following, where monkey is fed with electrodes, where they're recording from the cortex, prefrontal cortex, and the striatum. And the task is that the monkey has to look at random dot patterns, and the dot patterns are from one of the two prototypes. So it's a distorted version of either A or B. So it's either type A or type B. And monkey has no idea a priori. Monkey has not seen these patterns before, but monkey has to discover to distinguish between A and B and indicate which category it is by the sitar. So there's a queue for 600 milliseconds, and then there is a delay period for 1 second, where monkey is just waiting, and then there is a response where monkey has to tell whether it's A or B, and based on that, it gets a reward. So this is one trial, and it's trial after trial after trial. Monkey learns to differentiate the categories. It's a kind of reinforcement learning. And then they record the dynamics of the brain at the level of spiking neurons as well as the field potentials, and they also record the behavior. So in brain dynamics, the properties are the rhythms, the spectral properties, the phonies, and the actual spatiotemporal firing patterns. Whereas in cognition, there's a sensory encoding going on, there's reinforcement learning going on, there's decision making going on. So even if you know what category it is, monkey has to decide whether it's left or right. So it's a motor function. And then there's a working memory. So after a brief stimulus, there is a long delay where monkey has to hold on to that memory. So these are the cognitive functions going on, and the behavior, the accuracy improves, the response accuracy improves gradually, and it becomes like about 80 to 90% of accuracy. Then it almost gets every time it gets right. So our model also has to address all that. It has to address the brain dynamics, and it has to address the cognition. So we incorporate the receptor dynamics, where there are different neurotransmitters. There is GABA, there is AMPA, glutamate, NMDA and AMPA, and there is dopamine, there is acetylcholine. Then a very important aspect is the time course, the time scale. So each receptors have different time scales. Glutamate is very short, whereas GABA is quite long. So glutamate is about 10 milliseconds, 10 to 20 milliseconds, whereas GABA is 80 to 100 milliseconds. Then there's a plasticity going on. There's a learning. So the learning is happening at the level of synapses. And then the connectivity is accurate, as I said before. And then there's interaction between the neurons, assemblies, brain regions. And a quick kind of schematic look at what's exactly happening in a given trial. So in a given trial, there is a stimulus, visual stimulus. It goes to the visual cortex, it goes to the PFC, and there's a firing, initial firing, and then the activity goes downstream to the striatum. Now, the striatum 1 and striatum 2 are not two different regions within striatum. There are different populations of striatal neurons which are mapped onto the motor function. So this striatum, for example, controls the left saccade, controls the right saccade. So this mapping is very well timed. And we know that the downstream connections are geographically conserved. So these neurons will talk to these neurons, will talk to these neurons, and that will go back to the PFC. So what's happening is in striatum, there is a lateral inhibition happening, there's a competition happening, who will win. And that's why the decision making is almost like it's happening here in the striatum. So the activity, the initial activity, transient activity, which kind of starts the lateral inhibition. But what really tilts the balance of this competition is these TAN neurons, the acetylcholine. These are a very small population of neurons in striatum, but they're very important. They provide kind of noise which tilts the balance of which one will win. So, and it's observed experimentally that there's a between 100 to 200 milliseconds, there is a suppression of these TAN neurons. This is called TAN pause, and that's caused by another subset of striatal neurons, which is called striosome. So there's a matrizome and striosome, also called matrix and patch. They get cortical input. Both get very similar cortical input. So the matrizome is the main subset where the decision making happens, and what this does is it inhibits the TAN, and it also inhibits the substantia nigra, which is the dopamine. Both of these are important, and when that happens, about this time, the action selection has already happened, which means one of them has won. And then there's a suppression of dopamine activity in substantia nigra, which is somewhat corresponding to the reward prediction. So, as the learning goes, the reward prediction also modifies with time. And then the stimulus goes away, and for 1 second there is the loop activity. The activity persists in this loop, so that it's kind of working memory. And then there's an action, and then there's a reward, and based on the reward, there's a prediction error, which is the net change in the dopamine. Dopamine is released here. So the corticostriatal synapses are getting updated based on the dopamine. This is another computational primitive which I showed, the TAN circuit, just to get an idea what's happening. So the same, the striatum, the matrizome, they are getting the input, and the striatum is suppressing the TAN. So what's happening here is this is the activity. This is the activity due to the cortical input, and this is the activity due to TAN input. As the learning goes on, the TAN becomes lesser and lesser because this becomes more and more stronger, the striatum activity. So the noise becomes less and less, and then after a point, this becomes more dominant. So this is similar to the exploration versus exploitation in reinforcement learning. So initially, monkey is sampling a lot without getting stuck in one of the local minima, and then after some point, the noise becomes less, and the response will become more and more driven by what it knows. It's very similar to the simulated annealing approach. And the black curve here is the accuracy, which increases with time. So to summarize the results of this category learning, which is done by the circuit model, it can replicate a behavior, which means that it can reproduce the learning curve itself. And then at the level of dynamics also, not just the gross dynamics averaged over multiple trials, but even trial by trial dynamics is very nicely replicated by the model. So during the stimulus, the exemplar phase, where the stimulus is there in the cortex, this is from the empirical readings of the LFP, and this is a kind of estimated LFP from the spikes in the model. And then in the delay period, the activity slightly reduces, but it still exists. And something similar happens in the stratum as well. Now going further, not just one trial, but over many trials, what we see is that, so the early trials versus the late trials, if you look at the synchrony between the cortex and stratum, the corticostriatal synchrony, here it's measured by the phase clock value. In the beta band, so around 16 hertz, the synchrony is increased. Here you might see that there is an increased synchrony in the gamma band also, but the reason for that is that we have a very poor approximation of LFP, where we are just averaging the spikes. If we have a much better approximation, we might see a much better correspondence. The reason we did go for a much better estimation of LFP is because initially we were not even expecting anything to fit the data. It was just a sanity check. But even at the level of very crude approximation, we could see that a lot of things were being replicated. So this was again during exemplar. This is the increase, and during the delay, the increase is even much more pronounced. The increase in the synchrony, and that's what we see in the data as well. So during the exemplar, you see this increase in synchrony because of this synaptic. The weights of the synapses are increasing, and then there's this feedback, the thalamus to cortex, that's also increasing. And in the absence of stimulus, this is what is dominating. Furthermore, not just the synchrony of the corticostriatal connections, but if you go one level further of complexity, where you see that what's the difference between the synchrony in the correct trials versus incorrect trials. So take those trials where monkey gave the correct answers and those where monkey gave the incorrect answers, and you compare, is there any difference in the synchrony? So the difference between, so what you see here is the synchrony incorrect minus the synchrony in the correct trials. So if it's negative, means the correct trials have better synchrony. So in the empirical observation, they have reported that the correct trials, so which means the exemplar is on, and monkey has not even given the response, so monkey doesn't know whether it's correct or incorrect. There is some signature in the brain which is telling that it's going to be incorrect trial. Something similar we observed in the model as well, and that's not something we actually intended for. It came out of it, and this change in synchrony is more pronounced during exemplar phase, not the delay phase. So this made us curious, and we went slightly deeper. What's happening there? So we looked at the neural firing in the model. What's happening? And we observed that if you look at the cortical neurons in the model, after the learning, when the monkey becomes stable in its behavior, they develop a selectivity of neurons. So the neurons respond to specific type of stimuli more than the others. So half the neurons become tuned for category A and half the neurons get tuned to category B. But further, there is further subdivision that out of those, half of them are more active when the monkey is about to give the correct response, and half of them are more active when the monkey is about to give the incorrect response. So these neurons, so what you see here is neurons, each row is a neuron, and column is the type of trial. A trial with stimulus A, correct response, a trial with stimulus A, incorrect response, B, correct response, B, incorrect response. And then they are sorted in such a way that this set of neurons is more active when there's an A correct, this set of neurons is more active when there's A incorrect, and so on. So the existence of the incorrect neurons, which means these neurons which are more responsive at the first 200 milliseconds of stimulation, right? The stimulus appears, and within the first 200 milliseconds, these neurons are kind of indicating that this would be an incorrect choice. That's something we thought is, it has to be a model idiosyncrasy. I mean, there's no reason why it should happen like that in the brain. Now, a further statistical test also to see how significant this is, we did a statistical analysis where we said that we divide all the trials into two parts. One, in which we identify, so the neurons which indicate the incorrect responses, we call them incongruent neurons, ICNs. So in first half of the trials, we identify those ICNs and then use them for the next set of trials to predict at a trial by trial basis whether it's going to be an incorrect response or not, and how good they are at predicting that. So in the signal detection theory, you have this false positive rate and true positive rate. So this is all simulation, and you see that very nicely they predict that if it looks like it's going to give a wrong response, they actually give a wrong response. The surprising part is the same thing we found in the data as well. So they have the spiking data. So there are multiple levels of surprise there. First of all, why should it exist in the brain like that? And then the data set which they had, it's quite an old data set, but they didn't know such thing existed in the data. And they were very surprised when we told them that that's what happens. The data consisted of very few neurons recorded at a time, let's say 20 to 30 neurons in each session, which is very less. I mean, the fact that you can get any information from 20 neurons is from the PFC neurons, because so much is going on there. That itself is quite commendable, but in that, you get at least 3 to 4 neurons out of 20 neurons, which reliably tell you that monkey is going to make a mistake. And what you see here is from the analysis of those robust or like reliable neurons. So in the empirical trials also, we divided them into two. We identified the ICNs in the first half and then used that to see if they could predict the wrong trials beforehand. And this is an example of a single trial. If you could think of a thought experiment that if in the real time you're recording from, you're not recording from the cortex, the spikes, if you would actually identify those 3-4 neurons and use them as a predictor, and using that, you stop the experiment whenever monkey is going to make a mistake. You actually end up improving the performance of the monkey. So this is all the real data. So this experiment was not done exactly at that way. The experiment is already done, but from the data of the experiment, we asked that, what if we had stopped those wrong trials beforehand? And this is in the final stage of learning. And already the performance is quite good, but still it gets better. And the increase is statistically very significant. Just based on these 3-4 neurons. So that much strong is that effect. The existence of ICN is that much real. Now, why it exists that way, we still don't know. Why they are ICNs? Why do we need such a neurons which signal an incoming wrong choice? And sometimes they cause the monkey to make the wrong choice. One of the hypothesis is that it could be because that makes the behavior more flexible. So for example, if one of the rewards are flipped, so if A becomes, if initially if A is left and B is right, if A becomes right and B becomes left, which means the environmental condition has changed, then monkey needs to adapt to the new environment very fast, and there probably these ICNs will be helpful to always venture out of the correct behavior and change the behavior. So this was like for that. So this was a discovery from the model, not just that it explained something. It actually discovered something which existed but we didn't know. And then you can actually use this to try and model the dysregulation in the brain. What happens when something breaks down? What happens if you shut down some receptors, or if you there's a lesion in some region. So for some of the toy predictions which you did, like what happens if you cut this connection? If you should disable the dopamine receptors. So this is what happens according to the model, that the behavior breaks down. It doesn't learn, like it's mentioned here. And if you see the response, it becomes more random. And then the activity also, the corticostriatal synapses kind of decay, and it's mostly the TAM activity which is dominating, and that's why you see random behavior.
Anand?
Now instead of, yeah.
You can hear me, right?
Sorry?
Can you hear me?
Yeah?
Yeah. The previous slide that you mentioned, where you showed neurons that reliably predict the wrong outcomes.
Yeah. Yeah.
So, is it like overfitting? The balance between fitting a data where you don't want to overfit. So even if there are neurons which reliably predict that you're going to make a mistake, maybe there is some value in not using that information in your action.
Yeah, that's what. So that's what we need some neurons to consistently make mistakes. Somehow the circuit wants to encode the possibility of mistake also. It's there so that monkey can make mistakes.
Also, is there any molecular signature as to how these neurons differ? For example, neurotransmitters or receptors?
No. So they just emerge out of the learning process. They are the same neurons. It's the same pool of neurons. And just based on their, so we did some analysis that the initial connectivity itself kind of makes some neurons more primed to become the ICNs later. There's already some bias, so that leads to that might lead to them becoming ICNs. So there's no they're not genetically different. It's just the connectivity, I think. Okay. So now, when you break stereosomes, so when you break the stereosomes, again the behavior breaks, but in a different way. Here, the behavior is not random. The behavior just gets locked into one of the responses, irrespective of the stimulus. Then, what happens if you disable the TAM neurons, the acetylcholine neurons? Again, very interesting. So it feels like initially the model is learning, but then again it breaks down and it falls onto one of the local minima and just keeps repeating the same behavior. So it shows how each of the components of the circuit is very important, and dysregulation of that can lead to breakdown of the behavior and the function. So yeah, so that's mostly about this what I want to talk about. That a model which is predictive and mechanistic should incorporate the multiple scales and their interrelationships. And we have shown that it's possible to make a model like that with biological constraints and from physiology and anatomy, and not to overfit them for one particular experiment. So what we did here was we did not try to do any machine learning or to very high parameter fitting so that our model replicates the data. Very basic assumptions which we took about the synapses and the synaptic weights and the density, which again mostly from the literature, and the most of the behavior emerged out of it. So yeah, so the test case of category learning in particular, it could replicate the brain function at multiple scales, at the behavioral scale, at the dynamical scale of population dynamics, and at the neural firing. So it the spatial temporal firing is where most of the representation happens of the stimulus and of the decision. And it also could capture the subtle changes in the large scale dynamics. So the behavioral dependent, behavioral state dependent, learning dependent, all those dynamics was captured. And it also discovered a novel neural code. It suggested that incoherent neurons exist, and then when we looked at the data, they actually existed. So this kind of approach is very useful, not just as an academic advancement, but it can actually have applications to model different brain dysregulations, and especially to model the drug intervention. It's a very important problem nowadays because lots of pharma companies, they stay away from neuropharma just for this reason, because they don't have a good model to simulate what happens when the drug hits their targets. And that's why lots of pharma tests fail at phase 2 and phase 3, the clinical tests. So that's where we're going next. The idea is to make a library of models where we can actually say something about the drug intervention. So the drugs attack at this level, at the level of receptors. Some agonizes them, some antagonizes them, and that has an effect, large scale effect. It can make a conscious person unconscious. It can treat depression. It can treat bipolar, and so on. So yeah, these are my collaborators from MIT and Stony Brook, and Richard Ranger is my postdoc supervisor at Dartmouth. So yeah, that's where I'd like to stop. Thank you.